Hi there, welcome back to my channel Xgene and proteinX. In this tutorial video, I'm going to talk about types of CRISPR-Cas and molecular mechanism of CRISPR-Cas9. So in my previous video, I discussed about the mechanism of the CRISPR-Cas9, that is a simple mechanism. So before going to the main discussion, so I would like to have a quick recap that the basic mechanism of the CRISPR-Cas. So CRISPR-Cas actually is the immune mechanism of the bacteria against phage virus. So you can say also the defense mechanism of the bacteria. So in the CRISPR-Cas9 system, it has two different phases, acquisition phase and the interference phase. So in the acquisition phase, initially when the phage virus it attack any bacteria, then it incorporates its genomic DNA into the bacterial cell. So then some specific sequence is copied from there, and then it assembled in the CRISPR locus. So that specific sequence that is copied from the genomic DNA, that is called the spacer sequence. So in the CRISPR-Cas locus, it contain the spacer sequence, as well as it contain also some repeated sequence, that is the palindromic sequence, and also the Cas genes, that is a nuclease enzyme that cleave the foreign DNA. So and then this CRISPR DNA is transcribed to the precursor CRISPR RNA, and then it processed to some small segment of the CRISPR RNA, and later on this CRISPR RNA it associate with the Cas9 system. So the virus when the second time it attack, and when the same genomic DNA it incorporate inside the bacterial cell, then that RNA that matches with their genomic DNA sequence that attach with that genomic DNA, and then Cas9 it detected that DNA RNA duplex, and then it cleave the foreign DNA in the PAM sequence. So this is the interference phase. Alright, so let's talk about the classification of the CRISPR-Cas. So there are different kind of the classification in the class 1 and class 2 mainly, and also in the class 1 also there are type 1, type 3 and type 4, and class 2 contain also type 2, type 5 and type 6. So based on the encoded effector proteins, class 1 CRISPR-Cas system work with multi-subunit effector complexes consisting of 4 to 7 Cas proteins. So this system is also it's widespread in bacteria and archaea, and including all hyperthermophilic bacteria, and also it is around 90% all of this CRISPR identified in this CRISPR-Cas loci. So rest of the 10%, that is the class 2. So class 1 contain around 90% and class 2 contain around 10%. So this class 2 it contain the single multidomain effector protein, and are found almost exclusively in bacteria, this Cas 2. So as you see in this figure that Cas 1 it express Cas 6, and here also RNase-3, different kind of expression system, and also the interference phase there are different kind of Cas proteins, that is a nuclease for the cleavage. And in the class 2 basically here the Cas9, Cas12 and Cas13. So this is the basic difference, and class 2 is mostly used for the gene editing system. So class 2 system are suitable for the genome editing technology. So what about the class 2 system? So class 2 system it contain different kind of the Cas proteins that is responsible for the cleavage of the DNA, foreign DNA, and class 2 contain Cas9, it is in the type 2, Cas12a, Cas12b, Cas13a and Cas13b. These are different kind of the Cas proteins, and their characteristics are different. but most commonly use Cas protein, that is the Cas9. So this Cas9 is actually the CRISPR RNA-dependent endonuclease. So the main role of this Cas9 is to cleave the foreign DNA. So also the type 2 contain also the tracer RNA, and this tracer RNA actually it is the transactivating CRISPR RNA, which might have the evolved from corresponding CRISPR, and this tracer RNA is essential for precursor CRISPR RNA processing and target recognition in the type 2 system. So as you see here in this figure, this is the tracer RNA. So tracer RNA is essential for CRISPR RNA pre-CRISPR RNA processing. So let's move on to the molecular mechanism of the CRISPR RNA and tracer RNA Cas9 complex. So in the bacteria, so this system, this CRISPR RNA, tracer RNA and Cas9, this complex, it formed, and then it work for the cleavage of the foreign DNA. So as you see here that this is the tracer RNA, and the Cas9 content, there are two different lobes, REC lobe, that is the recognition lobe, and NUC lobe, that is the nucleus lobe. And the nucleus lobe content also there are three different domain, HNH, RuvC and PI domain. So HNH, this domain, and RuvC, this domain, there are same kind of function. So mainly I can say I can start from the REC lobe. So the REC lobe function of this REC lobe is to just recognize the nucleic acid sequence of the foreign DNA. And HNH and RuvC, these two domains, it responsible for attaching the DNA and cleave the two different strands of the DNA. And PI domain, it basically it recognizes the PAM sequence, right? So then it forms the tracer RNA, CRISPR RNA and Cas9 complex. So then in the interference phase, when the viral DNA it second time attack the bacteria, and that viral DNA contains the same sequence, then first the CRISPR RNA that contain the sequence, that sequence matches with the viral DNA sequence, then it bind with that viral DNA, and this is the recognition lobe. So this is the Cas9 I say here, this is the recognition lobe. So first the recognition lobe, it recognize the sequence in the viral DNA that match with the CRISPR RNA, and then the another lobe, that is the NUC lobe, that is nucleus lobe, and nucleus lobe contain HNH and RuvC domain, and these two domain are responsible for cleaving two different strand. So HNH domain it cleave this strand, and RuvC domain it cleave the another strand. But before that, PI domain it attach with, and it find or recognize the PAM sequence. So PAM is actually 2 to 6 base pair long sequence that follows the DNA region targeted for cleavage by the CRISPR-Cas9 system. So PAM sequence it recognized by the PI domain, and then RuvC domain and HNH domain it cleave the two different strand of the viral DNA. So this is how this viral DNA it works. This CRISPR-Cas9 system it works against viral DNA. So this is the immune mechanism of bacteria against the phage virus. So this is all about the molecular